Hi and welcome to my OCR A-Level Biology revision session with me, Christine. So today's lesson I want to look at phagocytes, which is part of your communicable diseases section in Module 4. So let's take a step back though and look at Module 2 and remind yourselves that we have got stem cells which are multipotent that have the capacity to divide and produce cells within the body. Now the hematopoietic stem cells are found in the bone marrow. And when they go through mitotic division, they will then need to differentiate. And what we need to understand now is we're looking at this differentiation into a specific type of phagocyte. Now, the one that you need to know is called a neutrophil, and we can identify a neutrophil based on the fact that it's got this multi-lobed nucleus. So as it was differentiating, that was one part of that differentiation process, and we can identify that if we look at a blood smear. The other thing that's an identifying feature is that it contains this granular cytoplasm. Now, that's because it has many lysosomes. Now, lysosomes are membrane-bound organelle. They are found inside the neutrophil and they contain lysozymes, and these lysozymes are digestive enzymes. So what we need to understand is how do neutrophils carry out their role in the immune response? And they do that based on the fact that if a pathogen has got into the body, they are going to carry out a non-specific immune response. It's non-specific because it doesn't matter what the pathogen is. It could actually be dust. It could be cell debris. What's going to happen is the role of that phagocyte is to move towards, engulf, digest whatever that foreign object is inside the body. So let's look at this in the stages that it has. So let's start with how the phagocyte actually moves towards. So the phagocyte is attracted towards that pathogen. It's attracted by chemicals that pathogen is actually producing. Now remember, let's take a step back again and look at Module 2 here. How does a phagocyte actually move? Well, that's to do with the cytoskeleton. So the cytoskeleton allows for the cell to move towards the pathogen. Now what this cell is going to do is the cell is now going to recognize that this pathogen is a non-self cell. So this is a non-specific immune response because it doesn't know which of the pathogens it's encountering. All it knows is that it is not a self cell. So we need to be able to regulate that our phagocytes don't just destroy our cells inside our body, that they are only destroying things that are not meant to be present. So this recognition that this pathogen is non-self is important, so it will know to engulf and digest the debris. So what we then need to understand is that after the phagocyte has moved towards the pathogen, what's then going to happen is this engulfing process. Now, the engulfing of the pathogen is into what's known as a phagosome. So this is an example of endocytosis. So the cell, the phagocyte, the neutrophil has moved towards the pathogen and is now starting to put out these projections of itself to surround, so that what it can do is it can engulf this pathogen into what's known as a phagosome. So a phagosome is a very specific organelle now inside the phagocyte which contains the pathogen. So that is an example of endocytosis. So what's going to happen then is these lysosomes, these membrane-bound organelles inside the phagocyte which contain lysozymes are now going to move and fuse with that phagosome. Now, how do they move and fuse? Well, that again is down to the cytoskeleton. So this is an energy demanding process, and what's going to happen is when that lysosome fuses with the phagosome, what we now have is a phagolysosome. So this phagolysosome is going to allow for this fusion, and those lysozymes are now released into that phagolysosome. So those enzymes are now going to digest and destroy that pathogen. So it's important that the enzymes, those lysozymes, were contained within the lysosomes until they have fused with the phagosome. So what we're doing is we are ensuring that this digestion is only happening in a very controlled manner. So we want to only digest and destroy the pathogen which was engulfed into that phagosome. So the other thing that happens with the phagocytes is they actually will release cytokines. Now, cytokines are a cell signaling molecule. If a phagocyte has engulfed and digested a pathogen, there has been some form of invasion. So what it needs to do is to signal to other cells to move to that area. So the cytokines, these cell signaling molecules, will actually cause more phagocytes to move to the area. And the reason they do that is because they will move in a direction of where those chemicals are coming from. It's a form of chemotaxis where the cell is being guided in the direction of where the release has occurred. So the other thing we need to know is, although you need to understand that there are neutrophils, that are phagocytes that will do phagocytosis, there's another type of phagocyte which is called a macrophage. Now, a macrophage will do phagocytosis, the same process, moving, engulfing, digesting, but the macrophages actually do it in a longer process. And the reason it takes longer is because a macrophage will actually take the digested parts of the pathogen, specifically, they will take the pathogen's antigen, and they will combine it with its own glycoproteins. And what the macrophage will then do is it will create what's known as an MHC complex. Now that MHC complex will then be presented on the macrophage's surface membrane. Now this macrophage is now going to be called an antigen presenting cell. And that antigen presenting cell can now stimulate cells in this specific immune response. And I will look at the specific immune response in a whole different video. So do check that video out. So the other thing we need to note is that our phagocytes, now remember our phagocytes can be neutrophils and macrophages. What they also have the capacity to do is they have the capacity to recognize what's known as an opsonin. So opsonins are very specific chemicals which are going to tag our pathogens, making them more recognizable to the phagocyte. So how are they recognizable to the phagocyte? Well, the phagocyte actually has receptors on their surface which will bind with the opsonins. And if it binds with the opsonins, it therefore is recognizing that phagocytosis needs to occur. So anytime an opsonin will bind to these receptors on the phagocyte will then start to engulf and digest. So antibodies are a specific type of opsonin. So if you look at IgG and IgM, these have the strongest effect on the phagocytes because these are specific type of opsonins that will cause phagocytosis to occur. So do check out my video on antibodies, but these are specific type of antibodies which act as opsonins, those way of tagging the pathogen to make them more recognizable by the phagocytes. So if you like this video, then please do click on the like button and subscribe to my channel. Also do check out my revision platform, www.aiqchat.com.